Great. Thanks again, everybody. Good luck and have fun. I'm going to invite the attendees to join in 5, 4, 3, 2, and
Hello, and thank you for joining today's Cutting Edge conversation, where we're going to discuss the challenges around sourcing human biosamples for research. We appreciate you joining and looking forward to having a lively conversation amongst our esteemed panel and with you, our audience. We will begin today's webinar by introducing you to our speakers. I'll kick it off with a high-level overview of the importance of sourcing human samples safely, and then you'll hear a presentation from each of our panelists about a particular technology that offers a proposed solution to the various challenges surrounding human biospecimen sourcing. It's focused on the practical application, and rest assured, these speakers and their companies have enormous expertise and diverse teams that support their subject matter knowledge. We would love to hear from you throughout this presentation. So please submit your questions by using the Q&A function, which you should see at the bottom of your Zoom window with the large letters Q&A. Also, a reminder, if you're interested in getting in touch with any of the speakers after the webinar, their contact information will be available on the last slide, and you can always access each organization through the scientist.com marketplace. Now, we'll begin with introductions. Firstly, myself, my name is Maria Manico, and I will be your host today. I'm the category director responsible for human biological samples at scientist.com. I come from a background of human biological sample acquisition, both within the organ donation as well as the standard of care pathways, which were a direct result of my research interest in tissue engineering and decellularization. Now, let's meet our speakers and panelists. First, we have McLean Collins, who is the Senior Vice President of Global Accounts at Discovery Life Sciences. McLean has extensively supported Discovery Life Sciences FFP samples and histopathology services over his 9-year career with the company. Intimately involved with product development, product management, quality assurance, and strategic sales. McLean is Discovery's foremost expert on DLS's FFP capabilities and how to best utilize them to support discovery, translational, and clinical research projects for biopharma companies of all sizes. Next is Dr. Richard Kondo, with a research background in cardiovascular pathophysiology and electrophysiology. Dr. Kondo has published 12 peer-reviewed academic publications on the mechanisms of cardiac ischemia and arrhythmias. He is currently Director of Business Development at AnaBios, where he cultivates relationships with pharma and biotech companies that partner with AnaBios to assess compounds in the areas of pain, neuroscience, respiratory disease, heart failure, and cardiac safety. Prior to joining AnaBios, Dr. Kondo was North American Sales Manager for Sophion Biosciences, managing sales for automated pump clamp technology for ion channel drug discovery. He received his PhD in cellular biophysics from Boston University. And last but definitely not least, Kathy Miller, the Senior Director of Product Marketing for Personalized Medicine at BioIVT. After beginning her research career at the University of Michigan, Kathy received her PhD at the University of Louisville School of Medicine before completing her postdoctoral fellowship at the Wistar Institute and the University of Pennsylvania. With a career that spans over 20 years at the bench, as well as a director and global product manager, all involving the use of biospecimens in the fields of cancer, immunology, and virology, Kathy is now Senior Director of Product Marketing, Personalized Medicine at BioIVT. Thank you, panelists, and thank you, attendees, for joining today. I will now begin with introducing the current challenges surrounding sourcing human samples. As most of you know, one cannot innovate in the human disease space without access to human biological samples, or HBS, as you may hear me refer to them today. Healthy control, disease state samples, or even biosamples investigating the effect of treatments, all of them are invaluable tools in every step of the preclinical and clinical pathway, whether it's for biomarker identification, for treatment targets, or optimization for diagnostic methods, or whether it's just developing a better ex vivo tool to optimize findings before you move to clinical trials. Human biospecimens are the answer. This is evident by the continued growth of HBS demand within Scientist.com's clients. We have supported over 8,500 orders, resulting from over 10,000 requests, and supported 39 plus organizations globally to get the samples that they need. Often, logistical and compliance issues are the bottleneck to acquiring human biological samples. So the Scientist.com's goal has always been to accelerate this by removing these hurdles. The platform enables researchers to find and connect to expert suppliers under a unified contractual framework. It enables quick interactions and communications, all governed by confidentiality, and proactively addresses any ethical considerations that may be required when sourcing human biosamples. So, Scientist.com, by addressing all of the organizational challenges, centralizing and automating the policies and procedures required, the researchers can now focus on all the biological challenges in selecting the right samples for their research, whether this is genomics, proteomics, or even logistical considerations in terms of transport time and storage. And on that note, I will now pass this on to our panelists, which will each cover how they enable researchers to find the right sample. It is my pleasure to introduce our first speaker, Richard Kondo from AnaBios. Richard, over to you.
Good morning, and thank you, Maria, for your introduction. As Maria noted, my name is Richard Kondo, and I serve as Director of Business Development at AnaBios based in San Diego. Next slide, please. At AnaBios, we have a unique human-focused program to support drug development through the measurement of physiological responses in primary human tissues. The goal is to provide human translational data at the preclinical stage in order to acquire viable and functional human tissue in a robust manner. We have developed a set of innovative methods to recover human donor organs through a network of donor hospitals throughout the United States. The focus of human organ recovery are visceral major organs such as the human spinal cord, peripheral nerve, heart, lung, liver, and kidney. And we have developed a battery of ex vivo and cellular assays to probe the compound interaction and to provide functional information on the efficacy of safety of compounds under development in human tissue. These efforts have been recognized by national regulatory authorities. We have contracts with the US FDA to provide cardiac safety data on reference compounds and on human cardiac tissue to support the FDA's effort to have a database of comparative human data with which to judge the data quality of incoming IND submissions. The next slide. So major challenges in the use of human organs in drug research on a daily basis are ethical consent of their use, scale of recovery, and initiation of ischemic damage on them at the moment of organs are no longer perfused with oxygenated blood. To meet scale and to guarantee ethical consent, we work with US organ donor centers to recover viable organs weekly. Mitigation of ischemic injury is handled by body and organ cooling and minimization of transport time to the research laboratory. For AnaBios, the typical post-mortem interval is under 16 hours. However, this is not sufficient if the end goal is to have viable organs capable of normal physiological function. And so we've developed a methodology to mitigate metabolic and ionic homeostasis dysregulation of non-perfused tissue. As a consequence, we're able to use the incoming tissue for ex vivo assays or to dissociate the tissue for primary cell assays. All incoming donor tissue is accompanied by anonymized donor data that includes demographic information, clinical overview of disease status, and medication information. In addition to tissue from healthy donors, tissue from donors with target diseases of interest are available. Next slide, please. A major use of human organ tissue samples is transcriptomics. The methodology that we use to maintain the viability and physiological functionality of human organs preserves the tissue from RNA and protein degradation. As part of our quality control, we extract RNA from the incoming organs and from our archive and assess RNA quality. The RNA integrity number, or RIN score, is a measurement of the intactness of ribosomal RNA and hence of mRNA quality. For general RNA work, a RIN score of 7 is the threshold for good RNA quality. The RIN scores in our samples are always above 7, and the average RIN for each organ type is over 9. The graph indicates the average RIN score of organs recovered over the past several years. It's compared to the RIN scores for equivalent organ tissues published by another source. As one can note from the data, the AnaBios RIN scores consistently rank higher. This means that transcriptomic studies, whether for bulk or single-cell transcriptomics, can rely on high-quality RNA for their molecular studies. Next slide, please. AnaBios recovers human dorsal root ganglia, or DRG, several times weekly. The DRG line the spinal cord bilaterally and transmit sensory or pain signals to the spinal cord. At AnaBios, they're either preserved for molecular studies or dissociated into neuronal cultures. The image on the top left shows the retained quality of the DRG. It's a stained section of the human DRG produced by the Aida Rolla lab at NIH. Examination at higher magnification at the right reveals preserved cellular and subcellular structures. The lower left image shows a confocal image of the isolated neuronal cultures, revealing actin filaments and neuronal beta-3 tubulin in antibody-stained cultures. In live cultures, neurons are electrically active and maintain a similar electrophysiological profile as in vivo. AnaBios uses these cultures in-house to support electrophysiological studies for clients' pain programs. These DRGs are available from both normal and from donors with a specific pain or neuropathic conditions. The DRG levels corresponding to different parts of the dermatome can be chosen. In addition, other ganglia such as nodose ganglia and thoracic sympathetic ganglia are available. The DRG isolated neuronal cultures offer the opportunity to treat the neurons with a compound or therapeutic in a modality prior to fixation or freezing for molecular work. Next slide, please. In addition to DRG, AnaBios recovers spinal cord. Recovery of spinal cord such that it remains electrically active is a highly specialized procedure. The upper image shows a recovered spinal cord. The spinal cord segments can be identified by the vertebrae level. The spinal cord is viable and can be sliced by a vibrotome. These slices can be placed in culture for several weeks, retaining electrical activity. The lower left video shows the dorsal horn of a spinal cord slice loaded with fluorescent calcium dye. Spontaneous calcium transients caused by underlying spiking electrical activity are visible as on-off speckles in the video. These slices are used to measure the pharmacology of various compounds, such as bixoxygen, shown here on the right, a current clinical compound used for pain. These data demonstrate that the recovered spinal cord at AnaBios are of the highest viable quality. Currently, spinal cord slices are available to clients in the AnaBios local area. For clients outside of Southern California, frozen spinal cord segments are available to do molecular studies. Next slide, please. AnaBios recovers whole human heart weekly. At AnaBios, either the heart is dissected into cardiac substructures or subjected to enzymatic dissociation to isolate primary human cardiomyocytes, as shown on the left. Isolated atrial and ventricular cardiomyocytes retain their in vivo rod-shaped morphology and contract and relax normally when electrically paced by an external source. Dissected cardiac muscle is used for drug experiments where their action potentials or forces measured, and to provide pharmacological data as shown below. Heart tissue is available from normal donors or donors with cardiac disease, such as the heart failure or atrial fibrillation. Clinical parameters such as ejection fraction, whether or not the donor had hypertension, diabetes, or previous myocardial infarction are available. Isolated live cardiomyocytes are available to clients in the AnaBios local area. For clients outside this area, preserved, that is frozen or fixed, cardiomyocytes are available. As with spinal cord slices, this distinction is a consequence of the challenges of transporting live cardiomyocytes and neurons while maintaining cell health. We are working on procedures to provide shipment of live cardiomyocytes and neurons while maintaining their health. Next slide, please. AnaBios recovers whole human heart, lung regularly. These lungs are available from normal donors or donors with respiratory conditions. Each lung is accompanied by demographic information, respiratory history such as asthma or smoking or medication. Lung tissue can be preserved through snap freezing or fixation for molecular studies. In addition, AnaBios dissects live lung tissue blocks that are sliced by a vibratome into precision cut lung slices. Lung slices, in contrast to myocytes and neurons, can be cryopreserved for shipment and thawed at a client's site for culture. These slices can be cultured for a minimum of 1 week. The slices can be used to assay compounds for pharmacological studies, such as shown on the lower right, where airway contraction was measured in response to agonist addition. Or slices can be treated by inflammatory conditions and used to measure biomarkers. The lower left graph shows qPCR measurements of IL-6 production in response to lipopolysaccharide treatment. Next slide, please. So in summary, AnaBios Human Focus Program is geared to provide translational data to clients' drug development program. The procedures used to acquire human tissue for these studies offer clients the opportunity to get human tissue samples and primary cells for their molecular studies. Finally, I want to announce that AnaBios has opened a human brain recovery program and will be recovering intact brain for preserved tissue samples. This involves different recovery procedures to maintain brain viability, and we will make these types of samples as fixed or frozen available in the next few months. Thank you for your attention.
Thank you so much, Richard. That was excellent. And as someone who has experience in the organ donation pathway, I find this incredibly fascinating. Our next speaker is McLean Collins from Discovery Life Sciences. Over to you.
Well, thank you so much, and it's great to be here today, and really enjoyed that previous presentation. My name is McLean Collins. I'm the Senior Vice President of Global Accounts at Discovery Life Sciences, and today we'll be discussing our approach to delivering novel biomarker insights from NGS-characterized FFPE tissues. Next slide, please. So I think we've all been touched by disease, and specifically cancer. It is ubiquitous. It doesn't discriminate, and even as we continue to advance new therapies, there are a lot of suffering patients who have late stage or recurrent diseases, and that was really the mission and the vision that DLS was created around. And so, as we have grown from one company into 10 companies combined, we really have created our mission to accelerate R&D through precision medicine. So we know that 86% of clinical trials fail, but biomarkers can significantly increase the odds of success. More than half of current oncology trials include biomarkers, and many of those trials actually may have multiple biomarkers, including a multi-omic approach with IHC and next generation sequencing. Next slide, please. So what we believe really is that biospecimens, and particularly highly characterized specimens, can not only speed up discovery and translational research, but can really drive the strategy and the efficacy of drug development programs through the entire clinical pipeline. And that's what we built the company around. And specifically today, we'll talk about how we have taken the next step to characterize our FFPE tissues to accelerate that entire drug discovery process. Next slide, please. So as you all know very well, specimens are not a one-size-fits-all resource. Just listening to Richard's discussion previously, the collection of a human normal anatomy is completely different than the collection of a cancer specimen. There are also a lot of different formats and matrices of these specimens. And so we want to treat each patient and each specimen as unique. Those of us who procured these can appreciate the complexity involved in this process. So what we know is that we must preserve the specimen and be thoughtful upon collection for downstream use. And you can see here some of the samples that we have at Discovery Life Sciences as a biospecimen provider, including peripheral blood. You can go forward here. Blood derivatives, including PBMCs for immunoculturing or immunophenotyping, proceed. FFP, flash frozen, and dissociated viable tumor cells. Next slide, please. So today we want to focus on our FFP tissue blocks. These are one of the most commonly available sample types, both for the research community, also in the clinic, but really also one of the most challenging for genomics. We have one of the industry's leading biorepositories with over 31,000 pathology reviewed FFP blocks, and really a novel approach to getting the most information possible out of those blocks through a combination of pathology analysis, IHC staining, and molecular characterization through next generation sequencing, which we'll discuss today. Next slide, please. So DLS, Discovery Life Sciences, is a global leader in highly scaled biomarker analysis. And you can see here our flow cytometry services, including 10x single cell library prep and sequencing, our large scale sequencing platform, including 10 NovaSeqs for highly scaled, high throughput library sequencing, as well as our immunohistochemistry and multiplex platforms. The question we have, though, is how can we combine a biorepository with a highly scaled biomarker service offering to really accelerate drug development and accelerate the timelines for our top translational and clinical research clients. Next slide, please. So today's topic is going to be high quality data from clinical FFP specimens. Next slide. Specifically around RNA, we know IHC is a standard in the market, and we're seeing an increasing move in RUO, and in some cases, prospective decision-making with more targeted panels like Illumina's TSO 500 for cell-free DNA, as well as solid tumor analysis. But FFP continues to be a challenge for extracting high-quality nucleic acids, specifically RNA. Next slide, please. Our Hudson Alpha Discovery Genomics Division has developed a proprietary extraction protocol with significant increases in RNA quality yields versus standard industry protocols. You can see here in the chart on the left, the DV200 score, which is a measure of RNA quality for FFP, and in many cases a better measure than a REN score for FFP specifically, looks at the number of fragments of greater than 200 nucleotides in terms of intact RNA. What standard industry protocol delivered 8% of samples with DV200 scores greater than 20%, where the DLS optimized protocol delivered 100% of samples with DV200 scores greater than 20%. And the most important point here is that the downstream sequencing success rate is a direct result of the quality of the input. So with a standard isolation protocol, a 33% success rate, with an optimized protocol from DLS, a 95% success rate. This holds true in ideal sample cohorts, but what about less than ideal samples? Next slide, please. We tried to replicate one of the more challenging cohorts we could find. So looked at older samples greater than 20 years, a combination of biopsies and resections, tumor percentage as low as 25%, necrosis as high as 40%. And you can see here that the proprietary extraction methods still hold true, with 97% of samples yielding a DV200 of greater than 20%. More importantly, the quantity of RNA isolated was also significant enough for us to do a ribo-reduced sequencing approach, 100 million paired-in reads with an average data output of 25 gigabytes per sample. Next slide, please. So why does this matter to biospecimens? What we wanted to do is take the next step to isolate high-quality nucleic acid and to provide next-generation sequencing to our existing bank to accelerate and speed up the research timeline. We have QC'd a variety of formats, including our whole FFP bank, as well as matched FFP in plasma, matched FFP in dissociated tumor cells, to provide deep NGS characterization to be a one-stop shop for the majority of our translational and clinical research clients. Next slide, please. This is an example of our currently available inventory. We have over 400 standalone FFPE cases with 300 to 400 coming each month. A significant amount of viable cell inventory, including our FFP and plasma for companion diagnostics development. This is also across a variety of panels depending on need. So we developed a QIAGEN multimodal panel with 64 genes, Illumina TrueSight myeloid panel for hematological malignancies specifically, as well as the Thermo Fisher Oncomine and Illumina TSO 500 for a much broader, more robust DNA and RNA panel, including many highly sought after gene fusions. Next slide, please. More importantly, we have opened up our fully integrated biomarker services so that clients can select samples from our bank and choose the panel that's most appropriate for their research, as well as in many cases, allowed our clients to send samples that they've collected that are less than ideal sample types in FFPE to maximize the biological and the next-gen sequencing data available from those samples to increase the utility for their retrospective clinical trial analysis or for their future research programs. Next slide, please. So what does this mean for you? In summary, we've put this all together, the bioanalytical samples as well as the bioanalytical services to develop smarter cohorts and faster timelines. And what this means really is you can trust our quality. You know exactly what you're going to get. It's been pathology QC'd by a board certified pathologist, as well as the associated molecular characterization data. You have access to an ever expanding bank of samples. So what we have today will only grow in the future as novel biomarker programs are developed and expanded into the clinic. And lastly, you have insight into research goals. And so our really entire philosophy is that we want to make our clients as successful as possible and help them shine in their research programs so that we can get therapies to patients more quickly in the clinic. Next slide, please. You can find us on scientist.com, Discovery Life Sciences. We're proud to be an 8-year partner on the platform with a 5-star rating. We want to be as responsive as possible, as quickly as possible. And in addition to our e-commerce store, we have a sales team that are highly trained experts in both the samples and services that would be happy to talk to you specifically about your research needs. Thank you very much for your time. It's great to be here.
Thank you so much, McLean. And I know that with FFPs being the most stable and more utilized form of human samples, the work you do is definitely facilitating research all around. Our next speaker is Kathy Mueller from BioIVT. Kathy, over to you.
Thank you, Maria. Today we'll cover who BioIVT is, what unique biospecimen solutions we provide, and how you can now access our digital biobank. BioIVT has a 30-year history of linking pharma, biotech, diagnostics, as well as academic and government scientists to patients in their unique data-rich specimens in order to partner with you to develop the right solution to fit in your research workflow to positively impact treatment outcomes to improve human and animal health. As a global organization with 18 operating facilities with over 350 IRB-approved collection sites located around the world, including in the US, UK, EU, Asia, Africa, and South America. Our sites span a range of clinical operations from local doctors' offices to large health systems to our now 7 BioIVT-owned donor centers. BioIVT has brought together 12 organizations in order to provide you a single source for your biological matrix needs. You can find us on scientist.com as BioIVT or as the original company, whichever you prefer and are most comfortable with. Our collaborative relationship with our partners and collection sites allows us to work closely with them to ensure that human biological specimens are ethically sourced with the specific experimental needs required by our client researchers. BioIVT provides products across the spectrum of human and animal-derived specimens, including both normal and disease state biological matrices, various primary immune and immortalized cells, as well as our AdMetox-related products and services. Our experienced scientists and technicians ensure fit-for-purpose solutions to accelerate your development across the entire gamut from target to clinic and back. From customized processing to creation of slides and TMAs, to cohort mutation screening, to high quality bulk plasma offerings, we get what you need in the format you require. Our comprehensive hepatic portfolio provides the solutions researchers require to consistently meet their needs on time and within budget. Now I'll introduce our new digital portal and run through a quick demo on how to perform searches and find cohorts on the portal. With our new portal, you'll be able to search inventory, define and refine your cohort, export your research results for upload to scientist.com portal, save your search and run updates on them for newly added inventory, and access documentation such as C of A's, regulatory documentation, and product data sheets. We have recently launched the BioIVT digital portal to provide you with instant access to our personalized medicine biospecimens, such as oncology tissues or autoimmune biofluids. Now you can view our inventory, export results, save and repeat searches, and get pricing information. There are multiple ways to navigate the website. To find these specimens from our homepage, you can view the product menu or type terms in the search bar. Alternatively, from the disease focus page, you can select the major focus areas that match your research. You'll be able to select specific indications that you are interested in. You will now see the available specimens and diagnostic options. Once you select a product, you will land on the product page. Here you will see details about the biospecimen, as well as quality and other resources that are available. From here, you can click Browse Available Inventory, or you can select product criteria through the drop-downs on the product page, which will then pass through to the inventory search, jump-starting your search. Available inventory that matches your criteria you selected will be displayed. You can add and remove additional criteria to further refine the results, such as diagnosis indication, matrices, species, and more. You can filter by donor information, diagnosis confirmed by our resident pathologist, format, specific disease requirements, as well as specific mutation annotations and HLA haplotypes. Once you have found an interesting specimen, you can instantly view the blinded IRB, approval letter, and informed consent used for that specific collection. You can also view the Apereo full slide image before adding specimens to your cart. You can do this through the Apereo app and screen as you would like. The additional details section actually gives you more information about each donor, as well as NGS sequence material. View the click price by clicking on the See Price button in the right corner of the listing. You can also export the list of inventory reports, useful to upload your requested samples via scientist.com. If you want to add one of these samples to the cart, search for the specimen ID in the inventory search, and you'll be able to do that. As an alternative, add the specimens to a quote via click the Add to Quote button. You will now not have the ability to change the quantity on the quote because there is only one specimen with this specific criteria available in inventory. If you no longer want the specimen, you can click the X next to the quantity to remove it. Now you can continue by reviewing your information and hitting the Submit button. You will see a confirmation page once your quote has been submitted. You will receive your quote emailed to you shortly, which you can then upload as well to the scientist.com portal. With access to our specimen biobank, we've made searching inventory and requesting a quote easier and faster because we know your research can't wait. If you have any questions or need any help, reach out to our customer service team who will be happy to assist you. Sign up for your account today by visiting bioivt.com. So now what can you do with our portal? You can look at available inventory and identify cohorts refined by disease stage, primary resection organ, or specific medication or social histories. You can look at specific mutation screen specimens like G12C positive lung cancer specimens. Ready to ship hepatocytes, immune cells, and dissociated tumor cells are easily reviewed, as well as different collection types, including anticoagulants, collection containers, and match sets. Importantly, you'll be able to see IRB approval letter and informed consent for tissue specimens, blinded and ready for your review. And you can also find educational materials. For these additional resources, make sure to check out our website for information from our experts, from quality to white papers to case studies and more. BioIVT offers unique solutions to support your research and elevate your science. Partner with us. Thank you.
Thank you so much, Kathy. And thank you everyone for listening in. This concludes the presentation portion of this event. I would like to remind everyone to carry on sending questions using the Q&A panel. And we will try and answer as many of them as we can during this session. I will start off with a first question to Kathy from BioIVT, since you've just finished your own. Is the online portal replacing the current processes to request available tissue inventory?
It is not. We see it as an additional channel for our clients. We still have our entire force of sales team members with an expertise in this, our inside search engineers who refine our search and provide outputs. So this is just an additional channel for our clients.
Excellent, thank you. Richard, I am going to go for one for you next. Although if anyone else wants to answer this as well, please feel free to jump in. How common are limitations of specimens used pertaining to specific purposes? And is there any restrictions? How do you keep track of those restrictions, basically?
Right. Yeah, it's an important question because, of course, these tissue samples are coming from human donors. So tissue samples do have some restrictions. First of all, the donor samples are actually anonymized, but privacy needs to be maintained. So sequencing for the purpose of identifying the donor is restricted. And when one signs a contract with us on this, that's actually part of what the customer is accepting those conditions. The other condition is, that comes along with that is that these samples cannot be used in any way to create an immortalized cell line based on the genetic line of the donor. So those are the restrictions.
And that's a very up in everyone's mind these days, with immortalized cell lines being collected without consent, given the recent news of the HeLa cells, which makes it very, very important. I'll pass a very similar question actually to McLean as well as Kathy. I'll start with McLean. So how do you, what procedures do you have to ensure that patients' informed consent covers your buyer's purposes, and how do you keep track of that? Very similar question.
Yeah, it's a great question. I think probably like my colleagues or peers in the industry here on the panel, we source from a variety of hospitals, surgical sites, repositories, really in the U.S. and around the world. And so there are really two concerns there. First is the ethical concern of collecting the sample. The second is the quality concern, right? Was the sample collected and processed in a way that is suitable for downstream research? So there are two types of, at least for FFP samples, two types of consent. There's the waiver of consent research protocols. There's also the informed consent for Discovery Life Sciences. We have both. Ellen Kargas, who's been in quality and regulatory for over 30 years, is our SVP of Quality and Regulatory, and really ensures that all of our collection sites, prior to any engagement of tissue procurement, that we've followed all the IRB approved protocols, that we have all of the appropriate regulatory and ethical documentation in place, as well as ensuring the quality metrics of those protocols. So it depends on the clinical sites in terms of which IRB we're collecting under and whether it's an informed consent or waiver of consent protocol. But all of that documentation is in place to ensure that those samples are sourced ethically. In terms of the downstream use, all of our protocols, we try to allow for as much current and potential, newly developed exploratory research as possible. As Richard mentioned, the sequencing for identification of donors, not able to develop the cells into a commercially viable product with the exception of our GMP donor clinic. But really want to keep those protocols as open as possible for research so that our scientists are not restricted. And that's all part of our IRB documentation.
That's fantastic. Kathy, would you like to give us a little overview? Yeah.
Certainly. McLean answered, as I think most of your respected well-based biobanks will do is, most of our collections are done under either waiver of consent or informed consent. All of those are IRB approved, except for postmortem collections where informed consent IRB approval doesn't come into play. And then most companies will do in line with what is IRB approval. We do have a white paper that is questions to ask your biospecimen provider. It may be a good source. You should be able to get your informed consent or waiver of consent documentation prior to purchase if required. BioIVT allows you to do that. The tissue ones are already available online. You can view those before even purchasing. And if you need them for other cohorts that we have, we also will provide those prior to purchase.
Excellent. Thank you very much. And just as a little added note here, I would like to remind our audience essentially that the Scientist.com platform does proactively request those as part of the statement of work submission. So your supplier should be able to provide some visibility into any restrictions or the kind of language rather that is included into an informed consent template. Next, I will go to McLean. There is a bit of a practical question. Do you have FFPs with known MSI characterization?
Yes, yeah, that's a great question. Microsatellite instability is certainly a very prevalent biomarker in colorectal cancer, increasingly in pan cancer research. We do have MSI characterized samples, including gastric, endometrial, colorectal cancer, and now with the TSO 500, a few other indications, including MSI high, microsatellite stable, as well as some samples with combined orthogonal protein expression, so deficient mismatch repair protein staining or proficient mismatch repair protein staining. So we can provide a list of those samples upon request.
Fantastic. Another slightly technical question. This one is for Richard. What are the differences between primary cardiomyocytes and neurons and pluripotent stem cell derived cardiomyocytes and neurons from the same lineage?
Yeah, well, thank you, Maria. Yes, actually, we are going to ask about the issue of primary frequently, and so getting human tissue in the form of pluripotent stem cell lines, of course, have been available for years now. What's different in terms of what AnaBios does is, of course, we're sourcing from human directly. And while the stem cell derived or the iPS derived cells have the human genetic background, they actually don't have the same mature phenotype as actually cells and tissues that have been developed in a person. So that remains a significant distinction between using primary tissue and stem cell derived sources.
That is fantastic. Kathy, I have a bit of a futuristic question for you. Do you have solutions that would support AI development? I suppose not that futuristic considering how common artificial intelligence is these days, but here we are.
Yes, and, and actually it's a historical database that we have that actually provides the future development. So BioIVT, our Astrand human tissue repository has been in business for over 30 years now. And we actually have maintained a database full of all of the specimens that have come in and out of the bank. With the board certified path reviewed diagnosis and the associated H&E slide image, and then all of the clinical data that's been reviewed for that. So we have over 1 million specimens that we can submit to an AI integration. Trying to think of the other word. Sorry, I'm blanking on it. But all of that can be used for AI. There is information on medications, on treatment. We have outcomes on some of the cases in the bank. We will still have tissue for some of the cases, but the older cases, we've probably no longer have those tissues, but we do have the data and the H&E slide that can be useful in the AI business.
Absolutely. And just as a follow-on question, does the online portal represent your whole inventory? Or do you have samples that perhaps are not listed yet and it's always worth?
Yeah, we have a lot of samples that aren't actually on the digital portal inventory yet. It sort of depends on what type of sample is, what is and what isn't there. We are continuing to align the data from our different banks and push it towards the digital portal. It's really a system of aligning data fields to make it available. So if someone is interested in something and they don't see it there, so they should definitely reach out either through the portal or through scientist.com or directly, and we can provide them additional resources that we may have.
Fantastic. We're all well aware of the pain that takes for data to, QC and be uploaded and available and make sense. So, good luck with all of that. We have, I think, 2 more questions. If anyone else is still coming up with questions as part of these conversations, please carry on using the Q&A. We'll try to get to them. McLean, I believe this one is for you, I suppose. Can I buy next generation sequencing samples that aren't FFP, especially with a known ALK fusion?
Yeah, absolutely. We talked about FFP because of the challenge in the RNA extraction process and our Hudson Alpha discovery protocol, but certainly we understand the need for various applications of next-gen sequence samples, including dissociated tumor cells that are live viable single cell suspensions of the tumor microenvironment for either additional flow cytometry analysis or cell culturing, drug response identification, as well as PBMCs, for germline controls, other sample types, including bone marrow mononuclear cells. So yeah, we do have a lot of alternative sample types. It's a really interesting question about the ALK fusion. There are a lot of different clinical programs right now working on gene fusions. These are very rare in terms of their biological occurrence in the clinic. And so we do have these samples, but the prevalence is so low that they tend to move almost as quickly to a research program as they come into our biorepository. So I just encourage people interested in those rare fusions to reach out to their sales representative or to check back on a regular basis on our website.
Excellent. And one last one, I'm going to direct this one to you, Richard. Are you able to provide viable living tissue samples? Or are you're able to provide cardiomyocytes and slices? Do you also make the tissue as a whole available to customers? Is that something you do?
Well, in the form of the precision cut lung slices, yes, that's a tissue that we provide. With regard to the spinal cord, and that can be shipped globally, right? With regard to the spinal cord slices, that can be provided to those who are in the Southern California area, not currently outside, as a live sample. With respect to the heart, no, actually, the tissues are themselves are not shippable.
Excellent. we're looking forward to more technologies that will enable those living tissues to be shipped more globally. I know our client base has an active interest in this kind of fresh living tissue, so that would be very interesting. Excellent. I think we went through all the questions. So thank you all very much for joining us for today's presentation. If you have any additional questions or you would like to get in touch with our team directly or any of the presenters, that information is available on this slide. And the recording of this presentation will be sent to you by email within the next day. Thank you so much to our speakers for their involvement in these cutting edge conversations. And thank again all of you for joining us. And we hope you found the presentation helpful. Have a wonderful day.